been an interesting day, to say the least. So I'm going to talk today about growth defense trade-offs and how phytohormones, plant hormones, can have different roles in growth, both growth and defense, and how this will affect ultimately the agricultural quality of any crop we're growing. So we want our crops to grow well and also to be able to defend themselves well against pathogens, and that's not always the case. So in the lab, we're trying to sort of hack that circuit, I guess, to achieve the advantage in both these aspects of plant life. So what's this concept of the growth defense trade-off? So it's an acceptable paradigm, I guess, that plants that are growing won't be able to defend themselves that well when they're doing a growth spurt, or if they're being attacked by a pathogen and they're busy defending themselves, they won't be able to grow as well. And that concept is sort of based on the idea that the plant has limited resources, so it can either do one or the other, but not necessarily both at the same time. And of course, their ability to either defend themselves or grow will depend on, of course, if they're being attacked by a pathogen at any particular time, but also on environmental factors. And I'm not going to go into this too much, but the processes that are required for growth and for defense can be very different. So obviously that also makes sense then that there would be a situation where either one or the other would be favored. So when a plant is defending itself, we're thinking more about the biosynthesis of compounds that are required for the plant to defend itself, either compounds that have direct activity against pathogens that can be attacking the plant, or compounds that the plant requires to fortify itself during its interaction with the pathogen. And when we're thinking about growth, we're thinking about burning energy, about burning sugar and making more energy so that the plant can grow and expand. So, of course, plant hormones are involved in both growth and defense, as is the case in any organism, really. They're mediators of key signals in most processes, I guess, in plant life. And classically, they were sort of, I guess, divided. Does that work? No. Okay. 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 Yeah. All right. So classically, they were divided. Well, you can just see the slide. They were classically divided sort of into two groups. There's a group that's more thought of as involved in growth, which includes auxins and gibberellins and brassinosteroids and also cytokinins, which are conspicuously absent from this diagram. And we heard about these hormones a lot today, I guess, right? And on the other side, in the context of defense, we're thinking more of jasmonic acid and salicylic acid and ethylene also, which is, again, absent. So if you think about it, if we're talking about this shift that has to occur between development and defense, and it sometimes needs to occur quite rapidly when there's a pathogen that's all of a sudden present and attacking the plant, then even though certain plant hormones were classically defined as growth hormones and others were classically defined as defense hormones, it's not so surprising that now there are roles that are emerging for classical developmental hormones in defense, because the trade-off needs to be fast. And as we said before, the plant has limited resources and a limited toolbox to use in different processes, right? So this is an example for cytokinin, which is now, I guess, sort of established. It was for years a classical growth hormone or a developmental hormone, and now it's been sort of established also as a defense hormone. So, and here in the diagram you can see it's primarily, in its developmental functions, it's regulating mostly meristematic fate and preserving juvenility, preserving meristems, and regulating cell division, and also source sync relationships. And in its defensive roles, it's regulating defense, basically, against different types of pathogens, and it does so primarily through its effect on additional hormones. So my lab has been researching sort of the role of cytokinin in plant immunity and in defense against pathogens for a while now. And, okay, no. שחר, זה לא עובר, זה עובר במחשב ולא ב... לא? המסך תקוע, סורי. לא קורה כלום. דבר אינו מתרחש. חוץ מהצילומים. אין, לא, זה תקוע, זה לא קשור בכלל למחשב, הוא לא מדבר עם המחשב עכשיו. זה כאילו, אתם רואים את הריבועים האלה? יש ריבועים כאלה של קיפאון? לא, לא. זה תקוע לגמרי.
מחשב הרוס ממה שאמרת.
כן. אבל המחשב בסדר, זה לא המחשב, זה משהו בחיבור, לא?
למה? הוא נתקע? את מעבירה אותו קדימה והוא נתקע?
לא, המחשב תקין, המסך לא מגיב.
איפה אנחנו עכשיו? בזה?
לא, אנחנו... במצגת, לאן הגענו?
כן.
לזה.
זה לא מקרן, זה מהמחשב למסך.
לא. תצעקי את ה...
לא, אבל לפעמים לסגור ולחבות אליו בלי לפעמים זה לא אסור.
עצרתם לי את השעון, אה?
או, הנה שחר.
המסך קפא שם.
המזגן אבל עובד מעולה בחדר. 10 נקודות. עכשיו הוא יקרוץ. אוי, שחריקו.
הייתי בדיוק בשירותים. סתם.
כל אחד יותר סטנדאפיסט מהשני, אבל זה לא...
מה פעם הוא קרס לו?
אני מתמודדת מעולה עם סיטואציות מלחיצות. עובדה א'. So I think everybody now knows I published 2 papers on this topic. We can move on. No stone was left unturned in that respect. So cytokinin inhibits disease, right? So this is just an example. This is true for both exogenous and endogenous cytokinin. This is an example with exogenous cytokinin. So if you pre-treat tomato plants with exogenous cytokinin, and then you infect them with the gray mold pathogen Botrytis cinerea, you can see that there's a strong reduction in the disease after the cytokinin treatment. And even more interestingly, if you apply a drop, so in the bottom you see plants that express a green fluorescent protein when there's a cytokinin response happening in the cells where the protein is expressed. And you see when you first put a drop of Botrytis, when you first put a drop of Botrytis on the leaf, you can see that there's a cytokinin response localized to where the drop of Botrytis was placed. Afterwards, it's sort of the cytokinin response moves to the outer edges of the drop, and then subsequently the signal will radiate out into cells where there is actually no Botrytis yet. So the cytokinin response is activated in the plant by the pathogen. And another thing we found is that, so here the green protein is not a cytokinin responsive protein. Here it's a protein that's required for defense, a receptor that's required for defense. And in order for it to transmit defense signals, it has to be on endosomes, which are those little circular dots which are mostly visible on the right-hand side after cytokinin treatment. So you can see that if you pre-treat with cytokinin, the availability of this protein to signal defense signals increases. And that's, of course, concurrent with the inhibition of disease. So immunity increases, disease is reduced. And the last thing I'll say about this, about the connection of cytokinin to disease inhibition and defense responses is that we were interested in examining how cytokinin can affect the microbiome because, as I'm sure you all know, the microbiome is very important for defense in not only plants but in all organisms. So what we found is that cytokinin does affect the microbiome, and it causes the microbiome to be more defensive. And I'm just giving a small taste of this, but what happens is when you have either increased endogenous cytokinin or cytokinin sensitivity, or you pre-treat with cytokinin, you get changes in the microbiome, you get a more robust and predictable microbiome, and most importantly perhaps in the context of defense, you get an increase in the population of bacilli. And that's again with both endogenous cytokinin at the top and exogenous cytokinin treatment at the bottom. So, and of course, this is the proof that this microbiome is required then to transmit the cytokinin mediated defense signals. So at the bottom, that's just what I showed you before. If you pre-treat with cytokinin, you get inhibition of gray mold disease. And at the top, the same experiment was done in sterile conditions, and you can see that the difference between the two different samples is much smaller. So cytokinin still inhibits disease, but to a much lesser degree, and that's also quantified to the right. So cytokinin promotes pathogen resistance, and it does so by both increasing plant immunity and boosting defense signals within the plant, and also by promoting bacteria within the microbiome that increase plant immunity, promote plant defense. So as I said before, right, there's this hormone, cytokinin. It used to be considered primarily developmental. Now I guess we can say it's also considered as a defense hormone. So it has these two functionalities, but then if there's a trade-off between these two functionalities, what's controlling that trade-off, right? So one of the things that's controlling this trade-off is, again, not surprisingly, protein translation. So most of the functions within a cell, within a plant cell, within any cell, are executed by proteins. And different proteins then will control or will execute developmental functions or defensive functions. And the balance between these types of proteins is then what can control this trade-off. And in that context, I want to talk for a second about TOR. So TOR is a master translational regulator conserved from yeast through plants, humans, mammals, animals, whatever. It's a very conserved protein. And depending on who it's interacting with, it can dictate the translation of different types of proteins that can then execute particular functions. And here, of course, it's defined as growth promoting and growth inhibiting. You can read just growth inhibiting as defense promoting. It's the same thing. So two quick results. I think this is the most science-heavy slide. On the right, we have the role of TOR in defense. So here we have a lesion area of botrytis. This is a quantification of botrytis disease. In the gray, we have the control, which is just the normal disease levels you would expect in these genotypes. So we have the background line, the wild type M82, the high cytokinin IPT overexpressing line, and the low cytokinin CKX overexpressing line. And you can see that when you have high internal cytokinin, you get a reduction in disease, and when you have low internal cytokinin, you get an increase in disease. And this is, of course, what I told you before. But then if we downregulate, so you can't knock out TOR, that's lethal, but if we downregulate TOR using the virus system that Ben talked about in the morning, if you were there, then we just get a constant level of disease irrespective of what the background genotype is. And this, of course, means then that TOR is required to execute these signals coming from cytokinin. If you don't have TOR, it's meaningless. The level of cytokinin becomes meaningless in the context of disease. Then at the right, we have the same thing sort of in development. So I apologize, I didn't put a picture of the wild type tomato leaf here, but the first 4 genotypes are highly complex. They're mutants or transgenic lines that have a highly complex leaf, and they generate a lot more organs than normal. And you can see if you knock down TOR, you get a decrease towards the wild type. You get less organs. And the 2 last genotypes, all of these, by the way, have 1 genetic change from the wild type. In the last 2 genotypes, if you in the mutant, you get a very simple leaf where there's a strong decrease in the number of organs. But if you knock out TOR, you get an increase in the number of those organs, more towards the wild type. And this again means that TOR is required to execute these developmental functions, and without TOR, the cues that are coming from either the mutation or the transgene are muffled, to the very least. So this is, again, just what I was saying before. TOR, depending on which proteins it regulates the expression of, can then regulate the trade-off between development and defense. But of course, as I said in the beginning, agriculturally, we don't want this trade-off, right? We want plants that are both very well able to defend themselves, very, very viable and strong against pathogens, and that generate a lot of yield. So how do we do that? So the theory we have in the lab is that in certain cases, priming for defense can uncouple this trade-off and cause development and defense to work in the same direction, in the positive direction, and I hope I'll be able to show you that. So I want to talk about Trichoderma. So Trichoderma, of course, is a biocontrol agent. It's a symbiotic fungus or commensal fungus, depending on who you ask. And it's marketed under a lot of names and uses green pest and pathogen control, has been so for many, many years. And in that context, I'd like to take a minute to showcase Igal's work. So you've seen Igal here throughout the day. He was on the organizing committee, and I think he did a wonderful job. And he was sort of one of the pioneers of biocontrol research. And this is work that came out of Volcani. It's important to say that, of course. And if you think about it, pest control or pathogen control that's not harmful to the environment and not harmful to human health is something that has become more and more desirable and important as the years have gone by, but it was relevant back in the 80s when Igal started working on it. And here you have an example of so he developed, together with Makhtashim and collaborators and his group, a particular strain of the species Trichoderma hertzianum for biocontrol. You can see cotton seeds. In one of the modalities, they coated cotton seeds with the product, the Trichodex. And on the right, you can, I don't know if you're able to see it that well, but they were able to, in his group, to show that the Trichoderma doesn't have direct activity against botrytis. So all the biocontrol is based on processes that are occurring through the plant. So I'm not going to read all this, but Ika wrote a very interesting chapter about, a book chapter about how they developed Trichodex for marketing and sort of the pitfalls, and it's very, very interesting, so I highly recommend that you read that. And this is what Trichoderma does. So T39 is this strain of Trichoderma hertzianum. It was tested primarily, it was originally developed against botrytis, but it controls a lot of different pathogens and a lot of different hosts. And so this is just an example of botrytis in tomato, lettuce, and pepper. And I showed you before there's no direct activity of the Trichoderma against the botrytis. So another way that Ishel and his collaborators were able to show that this is through induced resistance in the plant, is that they applied the trichoderma to the soil and tested botrytis, which is a foliar pathogen, and found that there was a strong reduction, right? Not again. Oh. I give up. No. Oh. Ah, okay. Okay, thank you. So I had 16 minutes, honestly. It's not me. So the immunity was induced this is just an example of how this shows through immune assays that indeed trichoderma induces plant immunity through response to flagellin and through ethylene production in response to another elicitor. But at the same time, of course, as a lot of you probably know, trichoderma is also marketed as a growth inducer, as a plant growth promoting fungus, as something that's sort of like a fertilizer. You know, it makes the plants grow better, it makes them bigger. You can see some of the examples of green soil, right? And again, a lot of plant hosts, and you can see with the trichoderma on the right, the plant looks just so much better, right? And this is another quantification of that. So here in tomato, you can see that the harvest index increases and the yield, the weight of the tomatoes increases if you treat with trichoderma. The Brix is unchanged. This has been fun, guys. Should I just leave it out of presentation mode? Is that what's making it, yeah? Okay, I'm at the end anyway, I'm at the end. So I just wanted to show you that this is, again, the line where the green fluorescent protein is expressed when there's CK, a response to cytokinin signals, and you can see that trichoderma promotes growth by promoting cytokinin signaling. And then the thought was that, and I'm really at the end, the thought was that if trichoderma can promote both disease resistance and plant growth, then maybe these two processes are in fact coupled in the same direction and not in opposite directions, as one would sort of assume from this great growth defense trade-off, right? And so this is just what I showed you before. Trichoderma reduces the botrytis disease on the left and increases plant yield on the right. And so then what we did was we did the same assays in a mutant that doesn't respond to jasmonate. So JI is jasmonate insensitive, jasmonic acid insensitive. And jasmonic acid is needed for the response to trichoderma. So if you don't have jasmonic acid, trichoderma isn't able to control disease as well. This is on the right. So there's virtually no disease control when you don't have the ability to sense jasmonate. And then the same thing is true for the growth. So the background line senses the jasmonate and responds to the trichoderma with increased growth, but the jasmonate insensitive line doesn't. So then these two processes maybe are coupled in the same direction, and that's what we're thinking now with plant priming, that it's not just, you know, a way to have green biological control, you know, that's environmentally friendly, but maybe it's a way to hack this development defense trade-off and, you know, maybe market products that are aimed at both increased resistance and plant growth promotion. So that's it. That's my group. The work was primarily done by Rupali and Merav, and some of what I showed was done by Iftach and our funding sources. Thank you.
So our last speaker for this session is Danny Ishel. Danny Ishel is head of the Post-Harvest Science Department in Volcani. His research focus is mainly environmental effects on dormancy and apical dominance of plants using the potato plant as a model. Danny works mainly on sugar signaling. We heard a lot about sugar already. And metabolism, combining physiological studies with targeted genomics. His research combines basic science with developing new potato cultivars